Hi everyone. Thanks very much for tuning in virtually. Hopefully everything goes smoothly today. I'm not too tech savvy, but I'll do my best. So yes, my name is Vanessa Brown and I'm currently doing my PhD at the University of Western Australia and Kings Park Science. And before we get started, I just wanted to acknowledge the Whadjuk Noongar elders, past, present and emerging, as the traditional custodians of the land on which this webinar is being hosted. So what I'm going to do is I'm going to turn my little camera off and go focus in on my slides. So if you just bear with me, I will do that now. Okay. So just before we get started, if you wanted to ask any questions, by all means do so using the little question button. You can type it in as I'm talking, and then at the end of my presentation, I'll do my best to answer all of those questions. So basically, today I'm talking about improving the Banksia Woodlands through restoration with seed spaghetti, and I'll be covering some results from some trials that I've run through my PhD, as well as from my honours. So let's get started. When I think of the Banksia Woodlands, I think of some really cool, unique plants, things like the Banksia species, like the Banksia attenuata and the Banksia menziesii. And I also think of the really diverse understory that it holds, like the Anagoxanthus manglesii, or the kangaroo paw, and the Xantheria presii, commonly called the grass tree. Not only is it home to lots of really cool plants, it's also home to a lot of unique and colourful animals. Beautiful birds like the splendid fairy wren and the Carnaby's black cockatoo, and the little honey possum. And if you look at the map on the left-hand side, you can see the kind of yellow creamy colour, and that is just showing the historical extent of the Banksia Woodlands. So it basically used to extend from Jurien Bay all the way through Perth and then down past Busselton. Unfortunately, due to extensive clearing because of urban developments and mining, pine plantations and agriculture, that extent has been severely reduced. As you can see with the green on that map, it shows, that because of that, there is a lot less Banksia woodlands throughout the region now. So I just wanted to show you some photos of what some typical Banksia woodlands would look like before it's been touched. And basically you can see the overstory is dominated by those Banksia species, and that typical diverse understory, really beautiful. And then unfortunately, a lot of the time this is what it looks like afterwards. After it has been cleared, there's just a lot of sand and weeds, but there is hope. Through restoration, we aim to reverse that degradation and restore the Banksia woodlands to pretty much something that looks like this photo here. So when it comes to restoring the Banksia woodlands, we have three options. The preferred option is using topsoil, Unfortunately, topsoil is not always available in the quantity that we want to work with. A lot of the time we're having to restore landscape type scales. And so another option is to use tube stock, but this can be really resource intensive. When you think about having to raise these seedlings in a nursery, and then also the cost of having to plant it out at site, it can get pretty costly. And then there's also poor survival rates, not only post the first summer, but also post second summer as well. So the third option is to use seeds, which can be pretty handy because it can be done on a really large scale and can be quite cost efficient. But in saying that, our seeds in the Banksia Woodlands can be really expensive. So I gathered some data from Nindathana, which is one of the local seed suppliers, and I looked at 1 kilo of seeds of just some typical Banksia Woodland species. And for example, the Banksia ilicifolia worked out to be over $17,000 per kilo. The Banksia manzesii and Banksia attenuata worked out to be just over $4,200 per kilo. The Conostylis was again $4,200 per kilo. And then Anagoxanthus manglesii, the kangaroo paw, was $2,400 per kilo. So we're looking at a really big cost. And when we're thinking that we're putting in this much cost into buying these seeds, and then the fact that there can be really low survival rates, generally around the 10% mark for survival, it's not looking good. And one of the things that can be a barrier to restoration is weed invasion and weed competition. And things like the perennial veldtgrass for the Banksia Woodlands can wreak havoc on the site. It can do things like alter the soil nutrients, alter the vegetation structure, and reduce the growth and establishment of these native species. So generally, weeds need to be controlled or removed, and this is usually done through herbicide application. But these herbicides can also have a negative impact on our native species. So we can look at using a product called activated carbon, which can provide herbicide protection. It's basically made from a carbon source. So you can think of that as wood or things like coconut or chicken carcasses. It really varies. And this is burnt at really high temperatures. And then it goes through this third activation step. And that can be done through steam, which is physical activation, or chemicals, which is chemical activation. Now, there is an alternative to activated carbon called biochar, and it's very similar. It's also made from a carbon source, but what makes it different is it's burnt at really low temperatures compared to the activated carbon, and it doesn't have to go through that third activation step. And basically, the way it works is like a sponge. If you think about your typical kitchen sponge, it has all those little holes in that yellow material, and if you were to soak up water, all the water would go into those holes. Well, that's very similar to activated carbon and biochar in that they have pores, just like the holes in the sponge, and herbicide will get absorbed into those pores But unlike a sponge, if you were to squeeze it, all the water would come out. You can't squeeze the herbicide out of activated carbon and biochar. It gets locked up into those pores. So activated carbon is not new. They've been using it for herbicide protection in agriculture for a while now, and they used to apply it in these large bands along the furrow, as you can see in that photo. But unfortunately, what happens is that band is not only protecting the crop species, it's also protecting the weeds that are underneath that band. So it's working, but it's not doing the best job. And it's also quite costly because a lot of product is being used. So the question is then, how do we focus that protection just around the seed? And one option is to use seed enhancement technologies. So at this point, I just wanted to explain kind of like what seed enhancement technologies are. It's basically a post-harvest treatment which can improve germination, emergence and establishment, or it can protect the seeds, and that can be from things like predation or pathogens. It can also increase tolerance to environmental stress and, for example, drought or frost. And it can also help to improve precision delivery. So now we kind of know what seed enhancement technologies are all about. We can break it down into 4 categories. There is extruded pelleting, or as I like to call it, and as you'll see in the title of my presentation, seed spaghetti. And I will go into more detail with seed spaghetti later on in my talk. There's priming, which is the controlled hydration of seeds in either water or a solution or a solid matrix, which brings the seed to the point of germination, but then it stops it before that, so the seeds are ready to go out. There's also flash flaming, where rotating seeds are exposed to a flame, and this can help to remove things like hairs and awns, which can be painful when using grasses. And there's also coating, which is applying binders and powders to the seeds through a rotary coater or similar equipment. So this is where I put my chef's hat on again. And basically, when I make seed spaghetti, I follow a recipe. So firstly, I combine all the dry ingredients, and that could include sand and activated carbon, clays, and in this photo, in the front corner, I have the Jacksonia seeds, which I use in my field trial, which I'll talk about in a minute. Secondly, I mix all of that together in an industrial pasta machine. Next, I add the wet ingredients to make a dough-like material. Then I extrude this dough-like material through the die of the industrial pasta machine and cut it into short lengths. And lastly, voila, you have seed spaghetti, and you plant it out and you wait for those seeds to grow. So our colleague in the United States, he had already tested some seed spaghetti with activated carbon with a certain herbicide, and it was working. But we wanted to know, can we use less activated carbon than what he was using? So our colleague was using about 50% activated carbon in his recipe. So during my honours, I ran a proof of concept glasshouse trial where we modified that seed spaghetti, and we had one type of seed spaghetti which had less activated carbon, And then we had another type of seed spaghetti which had no activated carbon in it at all. And I also had a control, which was the non-pelleted seeds. And I better put in there that it was important to us that we found out, if we could use less activated carbon, because if we're using less of a product, we're saving costs, which is always a positive. We used a grass species in this trial for two reasons. One, because you don't have a lot of time in an honours trial, and secondly, this grass species was susceptible to the herbicide we chose called simazine. And simazine is basically a pre-emergent herbicide, which is like you apply it to the soil rather than a post-emergent herbicide, which gets applied directly to the plant. So this is some of the results that came out of my trial. And the y-axis of this graph is showing the percentage of mortality, or basically that's death. That's got your little sad face next to it. And then across the x-axis is time in days, and that runs from 18 days to 34. And the first thing that I'll point out is that there was 96% mortality for the control seeds. So that was the seeds that weren't in any seed spaghetti. So basically from this, we knew that the simazine was working. Underneath that, with 77% mortality, was the seed spaghetti with no activated carbon. So yeah, I mean, it was working a little bit. It was providing some protection. But down the bottom, with 22 to 38% mortality, was the seed spaghetti with the reduced quantity of activated carbon. So we found that yes, using less activated carbon than that 50% that our colleague was using still protected those grass seeds from simazine. So then I went on to start my PhD, and I wanted to know if these seed enhancements could also improve emergence. So I set up a big field trial out at Nangara with the Hanson Heidelberg Cement Group. As you can see, it's just north of Perth. And I had two sites. I had a post-mine site, so that's the site at the top on the left, and that was had sand extracted, but they'd finished mining that area. And down the bottom, you can see I had a post-pine site. So there used to be pines there, but then they'd cleared it and been using it as like a layover for the trucks for the mine. So that was definitely a highly modified site. And in this trial, we planted out approximately 40,000 Jacksonia fasciculata seeds. So Jacksonia fasciculata is commonly called the grey stinkwood, and it's a very common shrub found in the Banksia Woodlands. It's a very cute seedling which grows up. It's kind of like, I don't know, people don't think it's the cutest plant, but I have a bit of affection for it now because I've been using it in so many of my trials. And you can see why we planted out 40,000 seeds, and that is because I had a lot of different seed enhancements. So on the left, you can see the activated carbon seed spaghetti. I had 3 different versions of the seed spaghetti, and I also had an activated carbon coating. Then on the right, I had 3 different versions of biochar seed spaghetti, and I also included a control which had no enhancement applied to the seed. So this is just a little snippet of results from that trial. And this graph on the y-axis, it's showing emergence of the Jacksonia fasciculata, And on the x-axis, it's showing all those different seed enhancements. And I made it easier to look at by putting all the activated carbon enhancements in blue, or the biochar enhancements in green, and then the control is the orange bar. And this is just from the post-mine site. So basically you can see that red dashed line that goes across and meets the orange bar, the control. And you can see from this that generally the seed enhancements were not inhibiting emergence, and some of them did a little bit better than the control. There was only one, which was one version of the biochar seed spaghetti, which just didn't do quite as well. So what I didn't tell you in that massive big field trial was that I also applied some herbicides out there, but it didn't quite work out as planned. So I had to go and run a Glasshouse trial, because at this point I still was unsure whether biochar could actually provide any herbicide protection. There was definitely information out there that biochar could provide it, but it really varied. So I wanted to know for myself if this type of biochar that I had could actually provide that herbicide protection similar to the activated carbon. So you can see me in my PPE gear there, looking fabulous, getting ready to apply the simazine to my trays. So in this trial, I tested 3 different types of herbicide this time. I used Fusilade and glyphosate, or Roundup, which are commonly used in the Banksia Woodlands, as well as the simazine again. And I also had a control where no herbicide was applied. So this time I had just 2 versions of the activated carbon seed spaghetti, and the biochar, I broke it down into 2 different doses. I had a low dose and a high dose, because I was unsure whether I needed more of that biochar to provide that same amount of protection as the activated carbon. And then I also had a control where it was a seed with no enhancement applied. So firstly, we'll just concentrate on the graph on the left, which was when the Fusilade was applied. And on the y-axis is survival, the percentage of survival. And then along the x-axis is all those different seed enhancements. And again, I'm following that same colour system where the activated carbon seed enhancements are in blue. The biochar is still in green, but I went the light green for the low dose and the dark green for the high dose, and then the control is the orange bar. So zooming in on that Fusilade graph, you can see a couple of things going on. Firstly, I didn't circle it, but the control, or the orange bar, is not there at all. And that's because nothing survived when it was sprayed with Fusilade. I have circled in red there, a lot of those biochar seed enhancements really didn't do well and had very low survival rates. The activated carbon was slightly higher, but didn't really perform that well. And then to the right of that is the glyphosate graph, so where the trays were applied with glyphosate. And I've just put a red dashed line along so you can see where the control sits. And again, I've circled those biochar seed enhancements because a lot of the time they just weren't performing as well as the control and had lower survival rates than the control. And I mean, even in this one, there were some other things going on there, but I think overall you can see that the biochar certainly was not protecting as well as the activated carbon in most cases. And that's not to say that all biochar wouldn't protect, but certainly the type of biochar that we have in the lab was not working as well. So I probably have more questions than answers right now. So I'm going into another field trial next year, so I can hopefully answer those questions. And what I want to do is expand those seed enhancements so I can test it on a range of native species. So I've only tested it on Jacksonia fasciculata currently. So I'd like to be able to test it on Banksia attenuata, the Anagoxanthus manglesii, keep the Jacksonia fasciculata in there. Also Acacia pulchella, Eucalyptus toddtiana, and Gomphallobium tomentosum. And this time we're locating that trial up in Gingin. And that's so that we can actually apply the herbicides that we want to at the rate that we want to. We were restricted in Nangara for good reason. We're over the water mound there. So up in Gingin, it's a lot safer. And we want to test simazine, fusilade, and glyphosate. And hopefully we can see this time in the field whether these different types of seed enhancement technologies are going to provide protection to these native species. So thank you very much for listening to my talk today and attending in virtually. And thank you to all of the organisations who have supported me so far during my PhD, and all of my volunteers who have come out in the field, and to my supervisors as well. So I'm going to flick back to see myself. So if you bear with me for a second, I will do that. Okay, and I'll just bring up the questions. So I've got a question here from Debbie, and she is asking, how do you determine the quantities of each of the seed spaghetti ingredients? That's a really good question, Debbie, and a lot of it was based off our colleague from the United States. He'd already done a lot of work with the seed spaghetti, and he gave us the recipe which we could then modify. But during my honours, there was a lot of trial and error, where basically I'd start off with a recipe and trial it through the industrial pasta machine, and there's a lot of factors that are involved in terms of the dough has to be wet enough but not too wet, otherwise then it won't extrude through the dye, as well as it can't be really dry either, because then the auger will start heating up and you'll have issues that way. And I guess at that time I was also testing a couple of different ingredients that hadn't been tested before, and I had a lot of issues. So it definitely worked out to do more of a trial and error and see, you know, okay, I ended up with this amount of sand, for example, and this worked best in the recipe. Okay, so I have another question, and it's, do you pre-treat your seeds to help them emerge before you put them in your seed spaghetti? And well, for the Jacksonia fasciculata, yes, I treated for dormancy first. They have physical dormancy, so I had to treat them in hot water, so about 90 to 95 degrees Celsius for about 1 minute. And it would vary depending on what seeds you were using as well, depending on, yeah, if you do actually need to pre-treat them before you put them in the seed spaghetti. And I just had a comment which was awesome talk, such interesting work. So thank you very much for your comment. Now I know that there's a bit of a delay with the time, so bear with me while I just kind of sit here for a minute and just see if any more questions come through. Okay, well, I haven't gotten anything else yet, but look, if you would like to get in contact with me and talk about seed spaghetti or talk about restoration of the Banksia Woodlands, I would love that. So please do, and thank you again for listening to my talk.